Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Enabling Early AAV Characterization, the Role of Mass Photometry in Upstream Process Development. I'm Abby Pinchbeck, an editor at BioInsights, and joining me today is Jacintha Victoria, who will highlight how integrating mass photometry as an at-line tool can enable faster and scalable analysis of AAV samples, allowing teams to make informed decisions earlier in development. Now I'd like to introduce our presenter. Jacintha Victoria is a strategic marketing and analytical leader with over a decade of experience in the biotechnology and gene therapy sectors. As the market development manager at Refeyn Limited in Oxford, UK, she leads initiatives to drive the adoption of mass photometry across critical applications in cell and gene therapies, including AAV, LV, adenovirus, and RNA-based platforms. So without any further ado, I'll hand over to Jacintha to start her presentation today.
Thank you for the introduction. Good day all. I'm Jacintha Victoria, Market Development Manager for CGT Space with Refeyn. And for today's presentation, I would like to talk about criticality of early characterization of AAV product and how mass photometry plays a key role in facilitating it in upstream processing. As part of this presentation, I would like to touch on the different upstream cleanup protocols available in the market, case studies done by our industry partners where they couple these protocols together with mass photometry to enable data-driven decisions taken at the early stage in process development. Before we go into the AAV upstream process, I quickly want to touch on the significance of CQAs. CQAs or critical quality attributes are defined by regulatory agencies, and monitoring of these critical quality attributes are key because they ensure that your product is of the highest quality, is safe, efficacious, and it meets regulatory standards throughout the whole drug development process. From previous experience, we now know that failure to effectively manage your critical quality attributes can lead to clinical trial failures and safety concerns, including death. Here are some of the risks associated with poor control of CQAs, and these have been taken from case studies of analogous therapeutic molecules. And they include modified biodistribution, loss of therapeutic function of the product, and finally heightened safety concerns due to the presence of impurities and/or incorrect dosing. This gives an overview of the full AAV manufacturing process, and they're grouped into three phases: upstream, downstream, fill and finish. Upstream phase is where the production of AAVs happen. It includes culturing of the cells, transfection of the cells with the plasmid to initiate capsid assembly and packaging, and finally lysis of the cells to release the produced AAVs into the media. This follows through into downstream, where a number of steps work sequentially to help purify your product, deplete your impurities, enrich on your desired capsid, which is your full capsids containing your intact transgene, and finally buffer exchange the product into the formulation that's ideal for your product. At the fill and finish stage, your product at the desired concentration and dosage is filled into the container, and now it's ready for its intended purpose. The colored boxes below list out the different critical quality attributes that are monitored in the different phases in the process. One thing to note here is that purity is a key attribute that's monitored across all of the phases in the process. Now to focus on the upstream phase of process, here key activities happen in this phase during process development and optimization. Regardless of the drug, this phase usually starts off with cell line development and selection. This step is where you identify and even engineer your host cell line to optimize high AAV production and also good quality. This is followed through with the identification of the best media formulation to ensure that you have optimal cell growth, good productivity, and good yield. Plasmid optimization includes two parts. One is the sequence fine-tuning of the plasmid sequence, which is followed through with defining of the plasmid ratio. Having balanced plasmid ratio influences your capsid assembly, genome packaging efficiency, and overall the purity profile of the product. In the next stage, you're looking into optimization of your transfection conditions and also identifying the best strategy for lysis of your cells and harvest of your material from either the bioreactor or the shake flask. In this stage, you're screening a number of conditions, which are a combination of factors, reagents, and also different time points. Here, you're trying to pick the best or the most optimal condition or combination, which gives you maximized yield with no compromise on the purity or quality of the product. And at the final stage, you're looking at clarification and scale up, where you're trying to move your lab scale process into manufacturing scale, again with no compromise on productivity, yield, or purity. These are some of the roadblocks observed in upstream process, which prevents us from making informed and data-driven decisions for some of the key activities that we detailed in the previous slide. So in upstream workflow, one of the first challenges that you encounter is the presence of impurities, which interfere with your ability to accurately and reliably quantify vector yield and productivity. This makes it a challenge because you're not able to assess if the observed or measured titer reflects the true nature of your viable product. Because of the presence of immense amount of impurities in your sample, this can mask the true impact of process changes on your product, leading us to make suboptimal decisions, some of which might not even lead to improvement in product quality or purity, which was the intended purpose of this optimization. Analytical methods used in this early phase of the process struggle with high background noise coming from crude matrices. This limits your ability to characterize your product purity accurately, and this becomes an obstacle mainly when you're trying to understand the accurate purity profile of your product using empty full analysis. Crude samples carry inherent variability, which introduces inconsistencies which become very evident, especially when you're trying to scale up your process. Now, this affects your comparability between batches, and it makes it very difficult when you're trying to implement process controls for process monitoring and development, or when you're trying to replicate the performance at different scales. Unreliable data obtained from these impure samples not only causes a lag in the development, but also has significant regulatory consequences. Because of the rework and troubleshooting that's required in the downstream phase of the process, now you're looking at increased costs for production or development, a delayed timeline, and it also makes it even more harder to put a robust data package together to support regulatory filing and also process validation. As discussed in one of the previous slides, purity is a critical quality attribute that's monitored throughout the AAV process workflow. AAV, as we know, is a sought after tool for gene therapy products. An ideal AAV product should have high concentration of the desired capsid, which is your full capsid containing your intact transgene. But unfortunately, your undesired capsids, which are your empties with no transgene, and partial with truncated version of the transgene with or without residual impurities, co-purify with your full. This is why it's key to completely characterize your product at early stage in the process, because this helps reduce the risk of increase in cost for production and also delayed timelines where you're trying to rework your process and product in the downstream phase of the process. So how do you actually characterize your product, which is why you need right analytical solution. So your solution or the tool should give you accurate and reliable measurement of your different AAV populations, which are your empties, partials, fulls, and overfulls. It should be easily adaptable, should run at fast pace, and also should operate at a low cost. This is why we want to introduce our technology, which is mass photometry, as it checks all of the requirements to be the right analytical solution. SAMX is the mass photometer dedicated for AAV characterization. So mass photometry, as the name goes, measures the mass of your single particles in solution using light. As you can see from the image in the center, you have AAV particles in solution on a glass slide. And when illuminated with a laser, they scatter light, forming a contrast pattern, which in this case is a circular dot. Now the software takes the contrast information from each of these particles and converts it into mass using a mass calibrant, in this case mass reference P2. The image you see on the further right is the mass histogram generated by SAMX. So here, the software has taken the mass information from each of those particles and segregated it into different populations based on the variation in mass. The empty population is defined based on the known mass information for empty particles. The full population is defined based on the known transgene size of the product. The population that falls in between the two becomes your partial population. Although we now have a tool that can accurately and reliably measure your different AAV populations, is faster to adopt, is easier to run, and also cheaper to operate, this still encounters the same problems that we discussed in one of the previous slides when analyzing upstream samples, which is why we need quick purification protocols to help purify your AAV sample in the crude mix and also to deplete the sample of the impurities. In this section, we will discuss three different protocols and some data is generated by each of those protocols by our industry partners. The first protocol I would like to discuss today is the affinity ligand resin. These are nothing but polystyrene divinyl benzene beads ligated to AAVX antibodies. These affinity ligands are recombinant single domain antibody fragments targeting your capsid proteins on the AAV capsid. Looking at the schematic on the right, the first step is the incubation step, which takes about an hour, which is the time required for your AAV capsids to saturate the affinity ligands that are ligated to the resin. As you can see from the image, these ligands are not biased by the capsid populations, which means they can equally capture or bind to your empties, partials, fulls, and overfull capsid. After an hour of incubation, you now wash the beads to remove impurities and unbound particles. Finally, the AAV capsids are eluted from the affinity ligands using an acidic environment, which is then neutralized with 1 molar tris. A few specifications of this protocol. So the binding capacity of the resin is greater than 1E13 genome copies per ml of resin. The developer of this method is Généthon and it's called simple cleanup method. And the format that they used was a 24-well plate. As highlighted, this protocol requires an incubation time for about an hour, and automation is difficult for this, especially because it requires centrifugation or vacuum suction that to be incorporated into the automated workflow. So now let's look at some data generated using this protocol by our industry partners. This data was generated by our industry partner Généthon, where they utilized the affinity ligand resin protocol in a plate format and purified harvest material from bioreactor. The material pre and post purification were tested on mass photometry as seen on the right. So the clarified harvest, which is the pre-purified material, showed an abundance of impurities and no AAV populations when tested or analyzed on the mass photometer. Whereas post-purification, you now have examples from two different serotypes, AAV8 and 9, which shows that there is a significant depletion of the impurities and enrichment of your AAV populations. Again, this is very key because this helps you understand the true purity profile of your product at very early stage in the process. This way you can understand what impact your process changes are bringing to the product quality and also some optimization steps that need to be focused on to further improve on the purity, yield, and quality of the product at later stages in the process. This is a case study done by our industry partner Takeda, and the data snippet is from a publication by them. Here they utilize SAMX alongside AUC to understand if it can facilitate the selection of the best or optimal transfection condition for the AAV manufacturing process. Here they took AAV9 harvest material and purified using porous capture select AAV9 affinity resin, and then tested it on the MP, which is the darker shade of both colors on the bar chart, and AUC, which is the lighter shade of both colors. Green represents transfection mix A data, and red represents transfection mix B. As you can see from the bar chart on the left, both transfection mixes generated substantial amount of the undesired capsids, which are your empties and partials. Although that is the case, the purity profile obtained from transfection mix B was much better than that obtained from transfection mix A. This is solely interpreted from the percentage fulls calculated between the two transfection mixes. Again, why is this key or why is this significant? This is very critical because now you're able to understand the true impact of process changes, especially when you're screening different transfection conditions on the purity and quality of the product that it's generating. This helps researchers pick out the best or the optimal transfection condition and also understand key optimization points to further enhance product quality, purity, and productivity. This is yet another evidence to show that SAMX can reliably and accurately measure your different AAV populations, all in alignment with its industry standard AUC. This is the second protocol that I would like to discuss today, and it's the affinity ligand magnetic beads. These are nothing but Dyna beads, which are superparamagnetic spherical particles, which can be used for purification of any biological molecules as long as relevant affinity ligands are ligated to it. In this case, the Dyna beads are ligated to AAVX antibodies, the same ones that were used for the affinity resin that we discussed previously. So if you look at the schematic on the right, you have your beads, your magnetic beads ligated to the affinity ligands, which are equilibrated in the buffer, followed through with incubation with the sample. Again, incubation time here is very similar to the resin, which is about an hour, and this follows through with washing to remove impurities and unbound particles, finally elution in acidic solution and neutralization of the eluate in 1 molar tris. The binding capacity for this is about 5E10 to 1E12 viral particles per mg of the beads. Again, this can vary based on the sample volume and the plate format that you're going for. So the developer for this method is Rentschler Biopharma, and the method was called direct capture method. The format they used here was 24 to 48 well plate. To highlight again, incubation time is about an hour, but automation is possible here, unlike the resins, because a magnetic plate or a stand can be incorporated into the automated workflow. This data was obtained from another industry partner, Rentschler Biopharma, where they utilized the direct capture method in combination with SAMXMP to understand genome packaging dynamics of the cellular machinery. So there were 16 bioreactors running in parallel, where they took samples from each of them at regular 8-hour intervals and post direct capture purification tested it on qPCR ELISA and mass photometry to understand empty full ratios in each of those samples. qPCR ELISA data is not shown here, but what was observed was that high variability of roughly 30% was seen across the whole testing window. If you look at the graph on the right, you have the two shades of blue dots which represent full capsids again across the whole testing window coming from run 1 and run 2. Similarly, you have two shades of gray which represent partial capsids from run 1 and run 2, and the different shades of orange lines representing the trending of each of those capsids. So looking at the data, you can understand that partial capsids across the whole testing window showed very less variability. They were very consistent with about 5 to 10% difference. Whereas with full capsids, you saw an increase in the full capsid production at intermediate time points. So about 30 hours, you saw a 10% increase in the full capsid production. And at 50 hours, you saw about 20 to 25%. Notably, between 50 hours to 72 hours, which is the actual harvest time point, which is usually used for AAV products, the full capsid production was saturated. No notable changes or increase were observed in the full capsid production. Again, why is this very important? This gave insight for the researchers into the harvest time point, the genomic packaging efficiency, and the production of the different populations of AAV across different time points coming from different bioreactors. Again, this helps them understand what optimization needs to be done to further improve on this productivity and yield by choosing the right condition and time points. The final protocol that I would like to discuss here today is the cationic monolithic column. It is ion exchange chromatography, and these are nothing but porous polymethacrylate structures, when interconnected, form well-defined channels, as you can see on the image on the right. They help separate your biomolecules based on charge, and because these are cationic in nature, they readily bind with positively charged particles and repel negatively charged particles. And as you can see, these channel structures have more surface accessibility for AAVs or other biomolecules to bind to, therefore giving higher binding capacity. So the binding capacity for this product is about 1E12 to 4E13 viral particles per mil. The developer of the product is Sartorius Bioseparations and is available in a 96 well plate format. Like the previous protocols, the elution for the product is an acidic buffer followed through with neutralization in 1 molar tris. One key thing to note here is that unlike the affinity ligand resin or the magnetic bead protocols, which require an hour incubation for your AAVs to saturate the affinity ligand on the resin or beads, this protocol or this product does not require incubation, which saves you about an hour in the purification process. Automation is also available with this protocol. So this product has been available previously for downstream purification. The vendor has recently adapted it to upstream purification for upstream purposes. So stay tuned in for release of the protocol for you to adapt the product for upstream purification processes and a joint webinar where Refeyn and Bioseparations will be presenting together on the data generated to substantiate this claim. Now, to quickly summarize on the three protocols that we just discussed, so we have the affinity ligand magnetic beads, affinity ligand resin, and cation exchange monoliths. A few key factors for comparison. So, the incubation time is about an hour for both of the affinity ligand methods, whereas there's no incubation required for the cation exchange monolith. The sample volume is less than 2 ml for all of the methods, with the lowest being 1 ml requirement for the cation exchange monoliths. And finally, both your magnetic beads and cation exchange monoliths are automation compatible. Your affinity resin, it makes it harder to be incorporated in the automated workflow, mainly because of its requirement to either have a vacuum suction or a centrifugation step to be included in the purification process. All three protocols can be adapted to plate format to ensure high throughput workflows or to support high throughput workflows. Now that we've shown the significance of having reliable upstream purification processes in combination with mass photometry at early upstream process stages, now we would like to show evidence of how reliable and accurate those measurements are. This is a data snippet from a publication by Takeda, where they measured three different serotypes, AAV5, 8, and 9, on mass photometer, which is purple, against analytical ultracentrifugation, which is blue. As you can see, mass photometer identified four different populations in each of those samples: empties, partials, fulls, overfulls, regardless of the serotype, all in alignment with the orthogonal technique AUC, thus helping us conclude that SAMX is serotype agnostic and can reliably and accurately measure your different AAV populations, all in alignment with its industry standard AUC. There's a well-known lack of gene therapy standards in the industry, which causes inconsistencies in the ways assays are performed and how manufacturing processes are monitored across labs. So having appropriate gene therapy standards is key because it facilitates process validation and also helps keep track of batch-to-batch consistencies. US Pharmacopoeia has recently released their AAV empty and full standards, for which they've used mass photometry alongside other orthogonal techniques, which is SECMOS, CDMS, AUC, and solo VPE, to characterize their products. The data snippet below is from the Certificate of Analysis of the AAV8 full standard. The data shows that mass photometry reliably measured the full population, all in alignment with the orthogonal techniques in the industry. Mass photometry has been officially included in US Pharmacopoeia's general chapter for AAV manufacturing and QC guidelines. It has also been featured in their gene therapy analytical guide for AAV capsid content for both product and process characterization, and also as shown on the left for release and stability. So this inclusion reinforces that mass photometer is now a regulatory acknowledged tool for empty full analysis. This makes it a better and a stronger case for mass photometer to be adopted into every workflow for both characterization and QC release purposes. Let's look at the reliability and reproducibility of the measurements from mass photometer. For this, we took a sample and replicated it 18 times over three different operators. Then the mass and the empty full measurements from all of the replicates were calculated to obtain percentage CV or coefficient of variation. As you can see from the table below, the coefficient of variation for all of the measurements and replicates was less than 5%, helping us conclude that SAMX is yet a reliable and reproducible tool for empty full analysis. So to conclude, mass photometer is a single particle resolution tool, which is also serotype agnostic, and it can reliably measure your AAV populations, all in alignment with the industry standards. The highlight of this technology is its requirement to have low sample volume and titer to deliver high quality results. Its intuitive workflow can now make you have results within less than 5 minutes per sample, and it can be easily trained out to different personnel. With the GMP software release, it can now be adopted in both PD and GMP environments, thus helping us conclude that SAMX is indeed a comprehensive AAV characterization tool and has its significance in upstream process development. All can be done within minutes at a cheaper cost and also in an easier fashion. To introduce a portfolio, we have two mass photometers, 2MP, dedicated for proteins, antibodies, and nucleic acids, and SAMX for AAV characterization. They both have manual and automated workflows and cover different mass ranges as detailed below. Querétaro is the newest inclusion to our portfolio. It's called a macromass photometer because it can measure both mass and size of particles. It has been designed for larger viral vectors such as adenovirus, lenti's, and VLPs, and cover a size range of 40 to 150 nanometer. Thank you for listening in.